Well, good afternoon. So today I'm going to present some work we perform using Tara Ocean's data. And in contrast to most of the results you've heard so far, we didn't use any molecular data here, but we use plankton images instead. So I'm not going to go into the detail of this figure, because I guess you are all familiar with how oceanic ecosystem function. But I just want to use this picture to stress the critical role of large plankton in the ecosystem functioning, and to say that most of the knowledge on this large plankton comes from copepods, essentially. And this is illustrated here by these few quotes that basically shows that up to date, copepods are basically seen as the insect of the sea. But we all know that there is not only copepods in the zooplankton, right? So basically, if I try to review briefly some of the results we've seen today in the Tara Oceans from the Tara Ocean expedition, basically you've seen Colombo stock that Rhizaria, and more specifically the Chorodaria, are abundant and among the most diverse eukaryotes based on the barcodes, right? And in Year-on stock, basically the Rhizaria are a key component structuring the interaction networks. And then in the ASPIC, the Agulhas ring study, we've seen that the Chorodaria, one of the group that is actually affected by environmental change occurring in the rings. And finally, the talk that Lionel just gave is that the Chorodaria are one of the major taxonomic group that is correlated to the carbon flux, right? So just to illustrate that, here is just a couple of plots, just of pies, just showing the contribution of the different groups, this in terms of number of barcodes. So you see these Rhizaria, they actually contribute 18% of the V9 of the taxonomic barcodes. And if you zoom in, then just follow the blue here. If you zoom in this Rhizaria, most of them are actually polycysteines. And then if you zoom in the polycysteines, then most of them are actually the Chorodaria. So I guess most of you are not so familiar with the Rhizaria. So I'll just explain you in a few words their main features. So these Rhizaria are also known as sarcodines, usually by biological oceanographers, and they are composed essentially of the famous foraminiferas, the Radiolaria, but also Phaeodaria. And all these organisms are essentially skeleton-bearing heterotrophic protists, and some of them live in symbiosis with the microalgae. And all these Rhizaria, and specifically the Radiolaria, they do encompass a pretty broad size range that goes for the smaller ones down to 50 microns. And usually in the smaller end of this spectrum, you will find the Acantharia and the Spumellaria and Nacellaria, which are actually very important in the fossil records. So they are very much used in oceanography. And then towards the larger end of this spectrum, you will find the Chorodaria. And the Chorodaria are essentially skeletonless Radiolaria, and they do form large colonies of several cells, several hundreds of cells. And all the Chorodaria are photosymbiotic. They all live in symbiosis with the algae. And one of the characteristics, because of these large colonies that you have here in pictures, that these colonies are fragile. And they usually break upon collection with plankton nets, and whenever they would be collected, they would actually break apart with classical preservative fixation procedure like formaldehyde. So they basically are very elusive organisms, basically. And to study these organisms, in the work I'm presenting here, we decided to use an alternative approach that is not using molecular biology, but in situ imaging instead. So basically, this in situ imaging, the underwater vision profiler, consists in deploying a camera along a vertical profile and taking images of everything that passes through. So people, and using this tool, you basically can identify organisms that have a size larger than about 600 micrometers, half a millimeter. And previous studies by Lars actually have shown that on a global scale, so in different oceans, here you are the different oceans, that some organisms are abundant using this tool, but then it lacks taxonomic resolution. Here these groups are labeled like spine, star, sphere. And then on the other hand, there are local studies in Central North Pacific, for instance, that have identified colonial radiolaria as being highly abundant, an important contributor to the system. So what we did here in our study is that we relied on our taxonomic expertise of the Rhizaria groups to actually calibrate images from the UVP and to discriminate, and then we were able to discriminate between 10 different classes of organisms. And then we applied this improved classification to the images. And then for this study, we used the data from 11 expeditions. So one of them obviously is Star Ocean, that you can see here, the track. But we also use other expeditions, reaching a total of about 900 sampling stations and about 2 million images. And so from each of these images, we could assess the abundances of the different groups, right? Like by counting specimens. And then from each of the images, we could compute the biovolume, and then using carbon conversion factor from the literature, we could convert these abundances into biomass. So here on this first map, the most obvious thing is that basically the Rhizaria, they contribute about 1/3, well actually exactly 1/3 of the zooplankton as seen by the UVP, which is quite a substantial amount of organisms. And then what you can also observe is that these high contributions are essentially located in these intertropical regions, waters. And then by actually converting these abundances into biomass, and then comparing this biomass with the global data set from the literature that are a recent data set, published data set, then on the first 200 meters depth of the water column, we could estimate that the Rhizaria contribute for 5% of the global carbon standing stock, which was quite unexpected. I mean, this is a huge contribution for one single taxonomic group. And then if you look only at the meso and macrozooplankton biomass, I mean, the Rhizaria, they do contribute up to 36% on average, actually. And that was pretty impressive results. And then if you actually looked at the latitudinal distribution, so from north equators here and to south, and then this is the biomass, and then you compare the Rhizaria in blue, and then the copepods from these data sets. And then what you can see is that actually in this area of the oceans between like 45 north and about 20 south, I mean, the Rhizaria and the copepods, they have actually a pretty much similar contribution to the biomass, which was pretty unexpected. And then, because we did this improved classification, we were able to go a bit further into the analysis of this data set, and then we split this into 4 main categories. Here, so here again is a latitudinal distribution, and here are the 4 main groups that I'm going to talk about now. And this is a distribution on the surface layer of the ocean, between the surface and 100-meter depth. And what you can see here is the blue curve here corresponds to the Chorodaria, and this is biomass. And that this actually, the highest contributors are the Chorodaria in the surface layer, which somehow makes sense, because just to remind you that all the Chorodaria actually live in symbiosis with the microalgae. Just, I mean, this is emphasizing again the significance of biotic interactions in the structuring of the ecosystems. And if you look deeper, so this is between 100-meter depth and 500-meter depth. Actually, an interesting point is that this is not the Chorodaria anymore, but this is the Phaeodaria that contributes the most to the biomass. and these are strictly heterotrophic. And there is one interesting point here, is that these actually Feodaria, they are very, very abundant in the California current, where there is a huge peak. It corresponds to this data here. And this is, for instance, the contribution of the Feodaria and Colodaria in the global UVP dataset. And then this is without the California current. And it's actually a lot of the Feodaria in the total dataset contributes to are from the California current. And then when they actually form a layer at depth, and then we don't know anything about this layer, we don't know the function of this layer at depth in the current. And so just to try to summarize a little bit, the main results that I've shown is that first, that the large rhizaria, they do contribute at least to 5% of the global carbon standing stock in the upper 200 meters. That is a very significant results, I think. And that these rhizaria, they do contribute as much as the copepod biomass in the large oligotrophic intertropical oceans. And within these rhizaria, the photosynthetic Colodaria, these radiolarians, and then the heterotrophic Feodaria are the major components. And just to give you food for thought, that these data are actually very likely underestimated, as I remind you that we only looked at the larger size of the radiolarians, and there are a lot of small acantharians and small spumillarians and other radiolarians that are not taken into account in this figure. And then that basically that the rhizaria, they are not only diverse based on molecular data, but they do also contribute to the global plankton biomass. So basically, I'd like to finish by saying that I hope I convinced you that the rhizaria is a taxa worth attention, and that hopefully there is going to be more studies on this group later on. Thank you.